Okay, can you see the screen, the PPT?
Yeah.
Okay, so today's topic is about medical research methods. As I mentioned before, my name is Wang. I think this name is difficult for the foreigners to remember, so you can call me Maggie. And my English name is Maggie. So I comes from Chemical Biological Research Center. It's in School of Pharmaceutical Sciences in Wenzhou Medical University. As I show here, this is my email address. If you have any question about this course, you're welcome to send me any questions through the email. Okay. So I would like to introduce myself a little bit. I got my PhD, and now I'm the professor in Wenzhou Medical University. This is my email address. And here shows my education experience. First, I got my bachelor degree in Guangzhou and major in pharmacy. And then I got my master degree and PhD degree, major in pharmacology. And here shows some research experience of myself. I am the visiting professor in University of Los Angeles, and also I'm a research fellow once before in Harvard University, working on the project about immunology. Now I focus my study on inflammatory diseases, metabolic diseases, and tumor. So both of these diseases share the common is that inflammation plays important parts in these diseases. So I work on their anti-inflammatory compounds to block these diseases, to treat these diseases. So this is my research interest. And today I'm going to introduce about the medical research. First, I would like to talk about the concept of medical research. Okay, here shows the concept. Medical research is the basic research, applied research, or translational research that conducted to aim and support the development body of knowledge in the field of medicine. So as you can see here, medical researchers can divide it into basic research, applied research, and translational research. So what are these researchers? I will introduce one by one in the following slides. First, let's talk about their basic research. Basic research is the systematic study directed toward greater knowledge or understanding of the fundamental aspects of phenomena and is preferred without thought of practical ends. It results in general knowledge and understanding of nature and its law. So briefly, what's basic research about? It's kind of research to study the mechanism or the fundamental aspects of the phenomena in nature. Sometimes these studies have no idea if it can put into the practical use. For example, here shows a picture. This animal, I think all of you have ever seen this before. It's a snail. And also, as you look into this snail, you can see there are curves on their shell, right? If you monitor carefully, and then you can find this curve have two different directions. One of them are clockwise, another one is anti-clockwise. So some of the scientists set up basic research about this curve to see how to design their clockwise or anti-clockwise of this shell. So for this study of this project, it can know more about the fundamental aspects of this phenomena. However, I think there's no practical use for this project, right? But basic research is developed for know more about our nature, the laws and the regulation and the mechanisms of the phenomena. So this kind is basic research. On the other hand, is the applied research. So compared to the basic research, applied research is a form of systematic inquiry involving the practical application of science. In other words, it means that this kind of study you need to put into the practical use in the future. So it assesses and uses some part of the research community's accumulated theory, knowledge, methods, and techniques for a specific, often state business or client-driven purpose. So in brief words, it had to put into practical application in the future. Here shows an example. This is a production of monoclonal antibodies. Have you ever heard of this monoclonal antibodies before? So I think most of you know about the antibodies, right? When you immune the animal with antigen, and inside these animals, they will respond to the antigen and to produce antibodies. And usually the organ in our body to produce antibody is the spleen. And inside the spleen is usually the B lymphocytes, B cells, to produce antibodies. So for one of the applied research, we want to immune the mice with their antigen, and we want to produce antibodies. However, these B cells cannot proliferate forever in vitro. However, on the other hand, the tumor cells can proliferate or immortalize in vitro. So one project is want to produce the non-other proliferated B cells, which can be cultured in vitro. So how they do this project? They fuse these B cells with the tumor cells together, and then these hybrid cells, they can both produce antibody and also can be cultured in vitro for a long time. So these fusion cells can use for as the tubes to produce monoclonal antibodies. So for this kind of project, when we complete this research, and then we got different monoclonal single cells, and then we can culture the cells in vitro and then produce antibodies, which can be used back in clinic. So this kind of research is applied research. After the study, you can use the data or use the results to use in clinic or industry to produce products. So this kind is called applied research. And the last one is the translational medical research. Okay, translational medicine or translational research can also call translational science. It is a discipline within biomedical and public health research that aims to improve the health of individuals and the community by translating findings into diagnostic tools, medicines, procedures, policies, and education. So as you can see in this figure, usually the doctors in clinic find some questions. For example, they cannot treat their disease very well. Those patients do not respond to some kind of medicine. And then those doctors can raise questions from their clinical therapeutics, and then pass these questions into the labs. So in the labs, the scientists can design experiments to solve this problem. And after their experiments, they can do the data analysis, and then they get a conclusion which can feed back to clinic to solve the clinical problem. So this kind of research is translational medical research. Okay, so these researchers focus on iterative feedback loop between the basic and clinical research to accelerate knowledge translation from the bedside to bench and back again. So briefly, it raises questions from clinic and solve the questions in the lab with basic research, and then benefit back to the clinic. So this kind of research we call translational medical research. Translational research can be broken down into different stages. And also this translational medical research are very popular or very hot area for the researchers nowadays. As you can see, first, it can be developed or divided into two-stage model. The first stage is T1 stage. So T1 research refers to the bench to bedside enterprise of translating knowledge from the basic science into the development of new treatments, which means that bench is the basic research. When they get their conclusion from the lab and then bring this knowledge back to their clinic and then help to develop new treatments for the disease. So this is the T1 stage. And then the T2 stage research refers to translating the findings from clinical trials into everyday practice. So once it can solve, if it can solve their clinical problems well, and then it can put into their practical practice for every center to treat this disease. So this make it widespread in clinic. This is the T2 stage. So this one is the two-stage model for translational medical research. And next is the five-stage model, which is more well divided. The first one, T1, means the basic research study in the lab. And then T2, preclinical research. That's between their labs and the clinic. Then T3 is the clinical research. T4, clinical implementation. T5, implementation in the public health sphere. So I can give a very simple example. For my lab, we are working on the drug development. So once we will study in the lab with the mice or cells to work to evaluate if one of the compounds can be used for the treatment of cancer, for example. So if it works well in the animal or the cells in the T1 stage, and then it can move to the T2 stage. The T2 stage will evaluate the compound to see if it will work or not in human beings. For example, how about the pharmacodynamics of this compound? Are they toxic? Are they safe for the use in the human beings? And for the big animal model, for example, from the mice move to the primates, are they working, still working? So this is the preclinical research. If the data are very good, and then we can move to the T3 stage. We can apply for clinical trials. And then we will try these compounds in the healthy people first, and then their patients, and enlarge the number of the patients if it works well, and then it can get in the market to become a new drug, then move to the T4, clinical implementation. So this drug can be buy in the clinic or in the market. And if it works very well, and then we can transfer from our own country to all of the world for the international use. So this is the five-stage model, which is more detailed compared to the two-stage model. However, no matter what kind of model, this translational research focus on the problem found in the clinic, and then try to solve this problem in the lab with the basic research, and then transfer back to the clinic to benefit the patient. So this is the translational medical research. Okay, so here shows one more examples about the translational research. So here, this topic is about biomarkers. So what is biomarkers? A biomarker or biological marker is a measurable indicator of some biological state or condition. Biomarkers are often measured and evaluated to examine. We can use for normal biological process evaluation, or examine the pathogenic processes or pharmacological responses to a therapeutic intervention. So as you can see these photos, on the right up corner. What are the biomarkers can be used in clinic? It can be used for the prognosis. For example, we can find some biomarkers which have high levels in the blood compared to the cancer patient to the normal patient, normal healthy people. If they have high level of some kind of biomarker, it is likely that they have cancer in these patients. Nowadays, we already have many biomarkers for the cancer prognosis. For example, this CEA. So this is carcinoembryonic antigen, and also another one, AFP, alpha fetoprotein. So this kind of biomarkers you can test in the blood. If you have a higher level compared to the normal standard, then it is more likely that this patient may have the cancer in their body. So these biomarkers can be used for the prognosis of some kind of disease. And also biomarkers can be used for diagnostic. For example, specific biomarkers are related to specific cancer types. Here, this table shows some of the example. First, for example, breast cancer. So breast cancer, there are some specific biomarkers to help to diagnosis for the development of breast cancer. For example, the estrogen receptor, progesterone receptor, or HER2. So these three different biomarkers are very common biomarkers for breast cancer prognosis. And also for the colorectal cancer, EGFR and KRAS, and these are also popular biomarkers for the prognosis of colon cancer. And so this is for the prognosis and diagnosis of the disease, these biomarkers. And also some of the biomarkers can be used for the prediction of the disease. Some kind of biomarker is highly related to the therapy sensitivity of the disease. For example, some of the markers, if they have high level, it may be their optical, can help to choose the optical therapy, the best drug to treat them. So it can help to predict the effective of these drugs. And also, biomarkers can use for their pharmacodynamics analysis. It can answer the question, what's the optical dose for my body? So if they have a high level of biomarkers or low level of biomarkers, it can reflect that if this drug established well in our body or not. So it helps to adjust their optimal doses for the treatment of disease. And last, biomarkers can also be used for the prediction of the recurrences in their patients. If they have the high level of some of the biomarkers, it may predict that this cancer will return in these patients in the future. So as we have a summary here, we can use different techniques. For example, use the lab techniques or the bioinformatics techniques. Then we can find new biomarkers. Then these new biomarkers can be used for diagnosis, prognosis, and also some of the biomarkers can be used for the drug target to develop new drugs for the treatment of this disease. So this kind of biomarker study, we can call it translational medical research, because he finds biomarkers in the patient, and then based on this biomarker, the biomarker markers, we can benefit back for the treatment, for the diagnosis of this disease. So this is one example of the translational medical research. And here shows one examples for translational research. And this is our original article from the journal, American Journal of Translational Research, which is published in 2019. So if you are interested in this kind of study, after the class, you can download this manuscript for detailed information. Okay. And next, I would like to show one example in my own lab about the translational research. As you can see here, the first photo, we can see 3 obese people here. And as we all know that the obese in our body can cause chronic inflammation, and then this chronic inflammation can cause cardiovascular diseases in the patient. However, we don't know that how the fat cause inflammation in our body. We don't know the mechanism. So this question is raised from clinic. We know that fat can cause cardiovascular diseases, which is related to chronic inflammation. But we don't know the detailed relationship between the fat inflammation and cardiovascular diseases. So raise this question, and then we go back to the lab to study. And we can, for example, to develop the high-fat model, mice model. And in this model, we use saturated fatty acids to develop the obese model, because in the high-fat people, the obese people, usually they have a high level of saturated fatty acids in their circulation blood. And in our body, the most common saturated fatty acid is palmitic acid, which I said use PA for abbreviation. So this PA, the palmitic acid, is one kind of free fatty acid, which is also the saturated fatty acid in our body. And then we find that this saturated fatty acid can bind directly to the myelite differentiation protein 2, which I label here the MD2 protein, which is a co-receptor of toll-like receptor 4. And then the activation of this signaling pathway can activate the download signaling pathway and then cause inflammation, cause the inflammatory cytokines expression in the heart, which can cause apoptosis, fibrosis, hypertrophy, and increase the reactive oxide species in the heart. All of this can injure our heart and cause cardiac dysfunction. So this is the mechanism we found in our basic research. Obesity, which is more specifically is the saturated fatty acid, can bind to MD2 and then cause inflammatory cytokines and cause organ injury. So not only in the heart, but also this obese can cause injury with the same mechanism in the kidney, in the blood vessel. So we published this, our findings in these journals. And then back to the clinic. So if we have compounds to block their inflammation in their obese people, and then they can block their organ injury in the patient. So in clinic, we try to use the anti-inflammation drugs to treat or to block their cardiac or the kidney injury in the obese people. So this is also one kind of translational medical research, which is performed in our lab. And one interesting finding is that we found that the unsaturated fatty acids, for example, the oil fish, it can block this saturated fatty acid harmful effect. So the company said some of the unsaturated fatty acids may help to maintain the health of our heart. So it makes sense to this point. Okay, although translational research is very, it's a hotspot for the study. However, it still faces some of the challenges and criticisms. The major criticism is that most of the scientists may think that basic research is more important because many good drugs, for example, the penicillin, antibiotics, and benzodiazepines are found from basic research. So they think basic research are more important. For example, the basic research about the biological facts to study their function and the structure of DNA help us to understand better about the DNA, and then based on that, they develop a lot of drugs targeted to DNA. So they think basic research is more important compared to the translational research. And also, a lot of translational research, they have came to fail in the study. For example, in the Alzheimer's patients, the doctors can find high level of alpha beta proteins in the patient's brain. However, when they get these findings back to the lab, they have a lot of drugs targeted to the alpha beta therapy, but all of them failed. So some of the scientists say that maybe translational research is not that useful because they have a high level of failure rate. And also they find that some of the studies from the translational research, other labs cannot repeat the same result. So they think that the widespread reproducibility of this translational research may decrease the value of this research. Then this is the challenge and criticisms of this translational research. And here shows some of the translational researchers facilities which are famous in all over the world. As you can see, you can find the Queensland University in Australia, a lot of institutes in the United States, and also the Monash University. So these universities have their good institutes for the translational research. If you are interested in this studies, you can search online to their website to read more details about the translational research. And also here shows some of the journals for translational medicine, and you can download the manuscripts from these journals to read more about that. Okay, next, I would like to introduce different types of medical researches. As you can see, there is a triangle here, shows different types of medical research. From the bottom is the in vitro research, and next is the animal research. And it can develop ideas, editorials, and opinions. So all this belongs to the preclinical research, or we can also call them background resources. And then the next level is the clinical reports, which include the case reports, case series. And then higher level on top of this triangle is the clinical research, including the case control studies, cohort studies, randomized control studies, or randomized control double-blind studies, and top is the systematic reviews or the meta-analysis. So all of these are different types of medical research. We will introduce them one by one. Okay. First is the in vitro research. I think most of the students have dealt with this kind of research in the lab class, right? So in vitro studies are performed with cells or biological molecules studied outside their normal biological context. Here shows the examples. First, if you work on their isolation, growth, or identification of microorganisms, so this is one kind of in vitro research. And if you work on the cell culture or tissue culture, it also belongs to the in vitro study. Next, if purification of molecules, for example, you work on the proteins, DNA, RNA, or something else. So this also belongs to in vitro study. Next, purification of subcellular components. For example, you work on the mitochondria or the ribosomes. So this also the in vitro research. And next is the extraction of cellular or subcellular extracts. For example, we extract the wheat germ or the reticulocyte extracts. And last, identification of the protein structure, the RNA structure, the DNA structure. All of these studies are examples for the in vitro studies. So when you talk about the in vitro study, what's the most important advantages for this in vitro study? I think this in vitro studies simplifies the system under study. For example, if I want to evaluate some of the compounds, if they have the anti-tumor effects. So if we use the animal model or human being clinical trial, we need to develop the tumor inside the animals first, and then treat them with the compounds and then evaluate the data, right? So it takes a long time to develop the tumor inside the animals. However, use the in vitro study is much simpler. For example, we can use the, picture shows the 96 well plate here. We can culture the tumor cells inside these wells, and then we can treat different wells with different compounds, and then it can evaluate the survival of the cells in each well. If the compounds inhibit the anti-tumor effect, the cells will die, right? Then use the proper machine. We can observe the survival of the cells here. If we have a darker color inside the well, means we have more cells there. The less darker means the cells are dead, so the color reduce. So based on the color, we can conclude that if these compounds can remove or kill the cancer cells or not. So use this simple system, we can evaluate the anti-tumor effect of the compounds very easy, right? Okay, so also the next advantage for this in vitro study is that human cells can be used directly. As we know that not every experiment we can perform on human beings, right? We need to get the ethical approval from the community to prove that this treatment or this therapy is safe for the patient and it do no harms to the patient. And then next we perform the human studies. However, if we use in vitro studies, the human cells can be used directly. For example, we can use the human cancer cells in this in vitro experiment, and then we can directly evaluate the anti-tumor effects of the new compounds. So it makes the experiments much easier. And next, in the in vitro testing, is also quite useful in the characterization of some specific absorption, distribution, metabolism, and expression processes taking place inside the living organisms, which means that we can use the cells rather than the whole animal to do the ADME evaluation. It makes the system much simple for the study. And then we can get the predictor further to confirm in the animals. So this is also another advantage. And last, in vitro methods are usually amenable to miniaturization and automation, making the study for the high throughput screening methods. For example, in this 96 well plates, we can evaluate more than 20 compounds together for one experiment. So it's very easy to develop the high throughput screening methods for the studies in the lab. So based on these advantages, the in vitro studies are very common in every lab. However, it also has its advantages. Sometimes it can be very challenging to extrapolate from the results of the in vitro work back to the biological of the intact organisms. What's that mean? It means that sometimes we can evaluate the compounds have very good anti-tumor effects. It works very well in the cells. However, when we use this compound inside the animal or inside the human beings, it has no effect because cells are much simpler compared to the animal body. So sometimes it works well in vitro, sometimes not very well in vivo. So it's the major disadvantage of the in vitro study. And so next level, although this compounds works very well in the cells or in vitro studies, we still need to use animal studies to confirm our finding. So next level is the animal research, also called animal testing. Okay, so which is also known as the animal experimentation, animal research, and in vivo testing or something else. It means the use of non-human animals in experiments. So as to pay attention to this concept, non-human animals. We all call those study animal researchers. Okay. Example, use invertebrates, the mice, rats, rabbits, birds, fish, frogs, and so on for the experiment. And it can be found in basic research, for example, the study of the genetics, developmental, biological, behavior studies. All these studies belong to the basic research that can use the animals in the study. And also animal studies can be also found in the applied researches. For example, the xenotransplantation, the drug testing, the toxicology research, and even for the industry, the cosmetic testing. All of this applied research can use this animal research. However, when we talk about the animal research, we need to pay attention to the care and use of animals. Here has the 3R regulation. So what's the 3Rs? What's the 3Rs? So the 3Rs are guiding principles for more ethical use of animals in testing, which is first described long time ago in 1959. So it's a long time ago. Also, what are 3Rs? The first R is replacement. Replacement means that the preferred use of non-animal methods over animal methods whenever it is possible to achieve the same scientific aims. For example, before we used the animals for the lab class, but now sometimes we can use the computer modeling to replace those experiments. So here shows an example. This photo I took in UK about 2 years ago. It's in Brighton University. So this is a robot for the lab class for the students major in pharmacy. So this robot can respond to different kinds of medicine. As you can see, there's a screen here to monitor its function of the heart. And also it can respond to any other treatment from the students. As you can see on top of the ceiling, there is a camera here. Usually one of the teacher will sit behind this camera and to monitor each student to treat this patient. And then this robot patients can respond from the machine, and also the teacher can talk from this robot to respond to the treatment of the students. So in this kind of robot study, then the students do not need to use animal for their fundamentally lab class anymore. Then use this robot can replace the animal in their lab class. So this is the first example, the replacement. Next, the second R is reduction. Reduction refers to methods that enabled researchers to obtain comparable levels of information from fewer animals or to obtain more information from the same number of animals. What does that mean? It means that if we can get the conclusion from, for example, 8 mice per group, then we don't need to use 20 mice per group. That we can reduce the actual number of animals inside in this lab performance. And also we can obtain more information from the same number of animals. What does that mean? For example, if I work on the diabetic complications, as we all know that the diabetes can induce the injury both in the heart, in the eye, and in the kidney, right? So when I developed the diabetic model in mice, then I can correct all of these eyes, heart, and kidney. So I can obtain more information from the same number of animals. All these studies share the same control group, the same diabetes model group, and then we can evaluate the treatment for the diabetic complications, including happened in the eyes, in the kidney, in the heart. So we can get more information from the same number of animals. That's reduction. And the last R is refinement, which means that we need to use the methods to alleviate or minimize the potential pain, suffering, or distress, and enhance animal farewell for the animal used. So when the animals are suffering, sometimes you need to sacrifice them to make them stop suffering. And also when we do the protocols, the non-invasive techniques are also the first choice for the animals. So this is the refinement, which means make the animals feel better. That's the refinement. So this is the 3 R's for animal use: replacement, reduction, and refinement. Okay. And then when we talk about the animal studies, there are some regulations. Animal testing regulations means laws or guidelines that permit or control the use of non-human animals for scientific experimentation. Usually, every institute or every university, they will have a community to regulate all this use of animal testing. In my university is the Institutional Animal Care and Use Community in Wenzhou Medical University. So the members in this community will review the research protocols and conduct evaluations for this animal testing. And if they approve for this protocol, then after that, we can start the experiment. Otherwise, we cannot do experiments before they review all the protocols. So this is the local regulations in each institute. And also, if you try to publish your data overseas, for example, you want to publish your data in the UK journals, and then the animal use, you need to make sure that all these protocols comply to the Animal Act 1986 in UK. Although it's a long time ago, this act has a long history, but now still in use. So when you publish manuscript related to the animal use in the UK journals, make sure that all of the protocols follow to this Animal Act 1986. And similar in United States, we have another Animal Act, which is called Guide for the Care and Use of Laboratorial Animals, which is published for the National Academy of Sciences in the United States. So when you try to publish your animal data overseas, make sure that you have followed the Animal Act in those countries. Okay, here shows the example. This is the cut from the journals here. So when you do the experiments about the animal, so you need to show the detailed information of the mice. For example, how's the body weight? Here I'll set weighing 18 to 22 grams. So and also the gender of the animal, the male mice. And then the resources where you get these animals, we obtain these animals from the animal centers in our university. And also you need to mention the breeding condition of the mice. They fit in the animal room. They have the free access to the food and so on. So this is the detailed information of the mice. You need to mention clearly in your manuscript. And then, very important, you need to show the ethical approval of these animals. For example, in this paper, I mentioned all animal care and experimental procedures compliant with the national law, the local community law, police, and also the International Animal Act, and so on. Without those approval, most of the journals will reject your submission. So this one is very important. Write the ethical approval confirmation in this animal part of the paper. And then for the animal part, you also need to mention how many animals you use. Usually more than 6 and no more than 10. And then you need to mention how many repeated experiments you have done in your study. And some of the protocols, how to sacrifice the mice, are they under anesthesia, and so on. So if you want to know more about how to, most of the standard for animal experiments, here, this manuscript you can download later to see the requirement for the animal studies in most of the journals. So here shows different animal models in the lab. As you can see, most of the labs will use mice for their experiments because mice are easier to develop the gene knockout mice. So mice is the most popular model for most of the labs. And also rats are popular animal models in the labs too. Otherwise, some of the labs will use fruit flies. For example, in my university, some of the scientists will use the fruit flies to study the function of the eyes, the development of the eyes. So fruit flies is also the popular model for the study. And next is the zebra fish. It's also very common in labs because fish are cheap. And also we can monitor the blood vessel very easy. And so it makes it a very popular animal model in the lab too. And some of the models will use, the labs will use the rabbits and the non-human primates. But these non-human primates are usually very expensive. So most of the labs will use either mice and rats for their animal studies. So that's for the preclinical research, which includes in vitro study and animal research. Next, let's introduce about the clinical reports. So first is the case series and case reports. So case series and case reports consist either of collections of reports on the treatment of individual patients or of reports on the single patient. What does that mean? You can see the figures here. When the patients with some kind of disease go to the hospital, the doctors can take the chart notes, and if they find interesting cases, and then they can submit to the journal for the case reports or case series. So what's the differences between the case series and case report? A research for case series or case results may reveal information that will assist in a diagnosis. So when the doctors publish this data, it helps other people to diagnose the same disease. So the case series or case reports are very useful for the diagnosis. So what's the disadvantages for this case series and case report? Because they have no control group. As you see, only the patients come in the hospital and the doctors write down their symptoms, write down the treatments for this disease, and then submit to the journal, right? So all these studies, they have no healthy control group with which to compare outcomes. So case series and case reports have no statistical validity. So this is the major disadvantage of the case series and case reports. So what kind of disease or all patients can be write into a case reports or case series? It can be an unexpected association between diseases or symptoms. It can be an unexpected event in the course of observing or treating a patient. It can be findings that shed new light on the possible pathogenesis of a disease or an adverse effect or side effect. And it can be unique or rare features of a disease. And it can be the unique therapeutic approaches. It can be a positional or quantitative variation of the anatomical structures. So all these topics can write into case reports or case series. If only reporting one case, that is case report. If you are reporting several cases with the same symptom or same situation, then you can call it case series. So this is the topics or types of case report or case series. So here shows one example. It's about the multiple myeloma, which is misdiagnosis as the somatic symptom disorder. It's a case report, which means that it's only one case for this situation. It's a manuscript from the Frontier in Psychiatry. So if you are interested in this manuscript, and then later you can download it and read detail about this manuscript. So what's the roles of case report or case series in research and education? A case report is generally considered a type of anecdotal evidence. And also, case report and case series can facilitate recognition of new diseases and adverse effects of the treatments. And also, they can help understand the clinical spectrum of rare diseases, as well as the unusual presentations of common diseases. And they can help generate study hypotheses, including possible mechanisms of diseases. And last, case reports may also have a role to play in guiding the personalization of treatments in clinical practice. So this is the roles or the functions of case report or case series in research or clinical education. Okay, so we have finished the clinical report, which includes the case series and case reports. And next is the top level of the clinical research, including the case control studies, cohort studies, and the randomized control study, and the systematic reviews and meta-analysis. Let's introduce them one by one. The first one, case control studies. So as you can see, that case control studies are studies in which the patients who already have a certain condition and compare with people who do not. As you can see this figure, it's a study about the lung cancer patients, study the relationship between the lung cancer patients and smoking. So for example, we have two groups of patients come into the hospital. One have the lung cancer, one do not. So these two different groups of patients come to the hospital, and then the doctors can ask them, are you a smoker? how long have you smoked? How often do you smoke? So take the histories from these two groups of patients and then compare the history, and they can get a conclusion if smoking is related to the lung cancer development. So this is the case control study. So this different from the case series and case reports. It has a control group here. One is the lung cancer group, another one is the non-patient group. So these two groups have the control. Makes the case control studies more reliable compared to the case report or case series, but they are less reliable than either randomized control trials or cohort studies. So the control group selection, so the non-patient controls do not have to be in good health. For example, the patients, when we compare to the lung cancer patients, the control group would be the patients without lung cancer. They can have hypertension, they can have diabetes. They do not have to be totally good health. And also, controls should come from the same population as the cases. For example, if the lung cancer patients are Russian, and then the non-patient group should be the Russian too. If the patients are Chinese, and then the non-patient group should be Chinese too, then the controls should come from the same population as the cases. And their selection should be independent of the exposures of interest. So that means if you are studying the smoking and lung cancer, then either the patient or the non-patient, they can be a smoker or not smoker. So it's independent of the exposures of interest. And then the controls can carry the same disease as the experimental group. That means if the patient group have the grade 3 level of lung cancer, and then the controls can be the grade 1 level of the lung cancer. They can have the same diseases, but they need to have different grade or different severity, and therefore to bring different from the outcomes of the interest. So you can analysis if the smoking related to the severity of the lung cancer, then you can do this kind of study. So this results in a lower power to detection of the exposure effect. However, still can give some idea about your exposure factor in the disease. And usually, greater numbers in the study will increase the power of the study. Numbers of the cases and numbers of the controls do not have to be equal. Usually, the number of cases less than the numbers of the controls. Based on the period study, usually the controls versus the cases equals to 4:1 may be a cost-effective way to improve the study. Let's introduce the main advantages and weakness of case control studies. So this kind of study can be done very quickly, because when the patients come in, and then by asking them about their past histories, the doctors or the researchers can quickly discover effects that otherwise would take many years to show themselves. Which means that when the patients come in the hospital, they already have the lung cancer. You don't need to wait for a long time for the development of lung cancer. So based on these patients, you ask them questions, and then you can get a conclusion very quickly. And also, these case control studies are relatively inexpensive. Usually, we use the frequent epidemiological study, and then we can carry out by their small teams or individual researchers in single facilities, and then we can get conclusions. So because they don't need their specific equipments, we don't need the high the very difficult techniques, and you only need a few members to do the research, so they are relatively inexpensive. And next, researchers don't need special methods. They just ask the patients some questions and then analyze the data, and then they can get conclusion. So we don't need special methods for this kind of research. And the most important drawback in case control studies relates to the difficulty of obtaining reliable information about an individual's exposure status over time. For example, if you are working on smoking and lung cancer development, when the lung cancer patient come in the hospital, you ask them, how long have you been smokers? And maybe they cannot remember the exact correct number for 1 year, 2 years, 3 years, or 11 years. Sometimes they cannot remember very well. And also for the questions, how often for them to smoke, they maybe can remember last month I smoked 2 packs per day. But 10 years ago, maybe they cannot remember how often they smoked 10 years ago. So sometimes when you ask them questions, the data you get from them may not that reliable. So this is the major disadvantage for this case control studies. Last, the first study, however, although they have weakness for this case control studies, they are still the first studies to suggest a new medical conclusion. And for this first study, maybe later they can decide higher level of study to design another randomized control trial or cohort study to confirm the first conclusion you get from the case control studies. So usually for the clinical studies, the case control studies could be the first step to get the potential hypothesis, the possible mechanism of the diseases. They usually is the first step for the study. So although they have disadvantages, they are still very important in clinical studies. Okay. And also you need to pay attention that just because there is a statistical relationship between 2 conditions does not mean that one condition actually causes the other. So what does that mean? Here shows an example. For example, lung cancer rates are higher for people without a college education. Those people tend to smoke more. Does that mean that taking the high-level college education can reduce the development of lung cancer? Of course not. That does not mean that someone can reduce his or her cancer risk by getting a college education, right? So sometimes, although a higher education can conclude from a statistical relationship in 2 different groups, but this kind of data do not mean that one condition actually causes the other. So after the statistical analysis of your data, you need to have your own judgment to judge your own data to get the correct true conclusion of your study. Okay. Also, this is an example for the case control study. It's about the use of antihyperglycemic drugs and dementia risk. So after class, you can read this paper. And next is the cohort studies. Cohort studies. A cohort study is a study in which patients who presently have a certain condition or receive a particular treatment are followed over time and compared with another group who are not affected by the condition under investigation. So here shows an example. This figure shows an example. It's a cohort study about smoking and the development of lung cancer. Since a randomized control study to test the effect of smoking on health would be unethical, you cannot ask the healthy people, you smoke 2 packs per day for 10 years, and I want to know if you can develop lung cancer or not. It's unethical because we all know that smoking is harmful for the human beings, right? So it's unethical to do that kind of experiment. So now a reasonable alternative would be a study, the cohort studies. So for the cohort studies, you can find 2 groups of people. One group is smokers. They smoke by themselves. They are already smokers. And another group is the non-smokers. And then follow these 2 groups patients for a long time, for example, 5 years or 10 years. And then to see how many of them will develop lung cancer, and then compare these 2 groups and their smoking habits. And then you can get the conclusion if smoking is harmful to the patient, as is smoking related to the development of lung cancer. So this kind of study is cohort study. So cohort studies are not as reliable as randomized control studies. However, they are more reliable compared to the case control studies. It's a higher level of case control studies. Since 2 groups may differ in ways other than in the variable under study. So because why they are less reliable compared to the randomized control study, because they have other factors. We will describe that later in the next slide. Okay. And for the cohort studies, it can be divided into 2 types. One is the current cohort study. A current cohort study represents a true prospective study where the data concerning exposure are assembled period to the occurrence of the fact to the study. It's complicated. What does that mean? That means the figures here, the top figures here, one is the exposure. For example, are they smokers or non-smokers? So that's the exposure, the different factor, exposure factor. And then follow them for a long time to see if they can develop lung cancer or not. That's the current cohort study. Have the exposure first and divided them into 2 different groups and follow this patient to monitor if lung cancer developed or not. This is the current cohort study. And also another example is done by the Oxford Family Planning Association. They study the beneficial and harmful effects of different methods of contraception. So in this study, you have different groups choose different methods for the contraception, and then to follow the benefits, all the harmful for the side effects of these patients. So this is the current cohort study. And the next one is a little bit different. It's called a historical cohort study. In a historical cohort study, the data concerning the exposure and occurrence of a disease first. So, which means that we know they have different exposures. We collect the data of their development of the diseases, and then go back to separate the patients into different groups and do the analysis. That's a historic cohort study. They will get the disease first, and then back to the group division. And for example, the long-term mortality after the gastric bypass surgery. So here, the exposure is the gastric bypass surgery, right? And then the disease would be dead or survive. So that's the disease. And we collect the data from the gastric bypass surgery and monitor if they are dead or survive. And then back to divide it into different groups and then do the analysis. That's kind of go back study. So we call that a historical cohort study. So the main problem with the cohort studies can end up taking a very long time. For example, they do the smoking and then maybe follow for 10 years, maybe 20 years to get the development of cancer. So it may take a very, very long time to finish this experiment. And also, during this long time, things tend to change over the course of the study. Some people may die, they may move away, you cannot contact them anymore, or maybe they developed other conditions. For example, the smokers may develop their asthma, and maybe they have the hypertension, which may affect the development of lung cancer. And also during this long time, new and promising treatments may come into use. And all these factors can affect the really study of your, the conclusion of your study. And also, cohort studies are generally preferred to case control studies, since they involve far fewer statistical problems and generally produce more reliable answers, which means that cohort studies are more reliable or better than the case control studies. You can remember this. This is the comparison of the case control. Case control, they have to know their, they want to study the effect of exposure, for example, smoking. And then we know that they already developed the lung cancer when they go into hospital. And then back to us, if they are smoker, how long do they smoke? How often did they smoke in the past 10 years? And then get the results. That's the case control. And on the other hand, if you correct two different groups, one smoker, another non-smoker, and follow them to develop lung cancer and do the study, that's the cohort study. So that's the main difference between this cohort study versus the case control studies. So this is different. Okay, also this shows also an example for the cohort study, and you can download the paper after class. And next would be the randomized control studies. I think most of you have heard of the randomized control studies because it's very famous, very common in clinical trials. So these are for two groups. Usually we include a group of patients and then, and then randomized, divided into the treatment group and the control group. And then follow for a long time, follow for a specific time, sometimes a few months, sometimes a few years, and then compare the results of the study. Key data for this randomized control study is that you need to divide the patients randomly to all groups. So when you include a group of patients, randomly assign them to treatment or the control group. And then this randomly assigning can help to reduce the risk of bias and make the data more reliable in the study. So this is the randomized control study. Usually you first set up the standard to include the patient, and also you also set up the exclusion of the patient standard, and then input enough number of the patients, and then randomize to develop them into two different groups, one with the treatment, another one without the treatment, and then compare the outcomes of these two groups. And then you can get a conclusion if this treatment is effective or not. This is the randomized control study. So with certain research questions, the randomized control studies cannot be done for ethical reasons. For example, as we mentioned before, we want to study the smoking on health. You cannot ask someone to smoke 2 packs a day and another group to abstain smoking. So this kind of study, because of the ethical reasons, we only can do the cohort study. And randomized control trials are the standard method of answering questions about the effectiveness of different therapies. So if you are working on a therapy question, you want to know, for example, if penicillin is effective on the treatment of COVID-19, for example, then use this randomized control study. One group used the penicillin for the treatment, another one do not use this penicillin. Then compare these two groups, then you can know if it's effective or not. This is the standard method of answering questions about the effectiveness of therapies. But still, it has its disadvantages for this randomized control study. First is time and money. Usually takes more than $12 per candidate, so it's expensive. You need to have a large number size of patients, and you need the machines to evaluate the condition of the patients, and usually it takes years or decades for the study. And also conflict of interest dangers. Some studies are fully or partly funded by the healthcare industry, so they are more likely to want their positive results. So sometimes they will include the patients which are more, which is much easier to get the positive results. So this kind will cause the conflict of interest danger. And the ethics. Ethically, it may be necessary to obtain ethics approval from the patient. The patient needs to know what kind of trials are going to use in themselves, and you need to get their approval from the patient, get their agreement from the patient before you do the experiment. Otherwise, it's unethical studies. So, when you do the randomized control studies, you need to get the trial registration. In 2004, the International Community of Medical Journal Editors announced that all the randomized control studies, all the clinical trials, you need to register before you do the experiment. Otherwise, you cannot publish in the medical journals. This is the website for the clinical trials. You may get on this website to see what's the ongoing clinical trials in this website. It's very, it has many information about the clinical trials in this website. And for the double-blind studies, here the double-blind study is one in which neither the patient nor the physician knows whether the patient is receiving the treatment of interest or the control treatment, which means that the patient will be randomized, divided into two groups. One will receive the pills with the medicines, another one is only the placebo control. But neither the patient nor the doctor knows which group is getting the medicine, which group is getting the placebo. So this even higher level of the randomized control study, which is called double-blind randomized control study. So this double-blind study is the most rigorous clinical research design because it can eliminate the placebo effect of the study. Why we will have the placebo effect? The placebo means that we don't have the effective medicines inside the pill. However, some kind of placebo still also have as strong as the effect compared to the evaluated medicines, because the patient or the doctors have more optimistic about the new treatment. So if they know they are getting the new treatment, experimental treatment, they tend to get more positive results. So use this double-blind experiments can remove this placebo effect. Okay. So this is the example for the randomized control study. Later you can read them online. And last is the systematic reviews and meta-analysis. What's that mean? That means others already performed different studies. They all work on the same topic. And then based on the standard, you can combine all these studies into a combined big study that is called the meta-analysis. So a meta-analysis is a survey in which the results of all the included studies are similar enough statistically that the results are combined and analyzed as they were one study. Briefly, it's others perform different experiments on the same topic. Then, after analyze, you can combine combine some of them into a big study to get a more reliable result. So in general, a good systematic review or meta-analysis will be a better guide to practice than an individual article because they have different model to combine different experiments. So they are supposed to be more reliable for this field. This is the systematic review and meta-analysis. The main stages of the systematic reviews are, first, you need to have a question, a clinical question. So define a question and agreed and objective method. And then research for relevant data. So search in the database to collect the suitable research for the combination later. And then a search for the relevant data. And then extraction of relevant data. This can include how the research was done, who participated in the research, how it was paid for, and what happened, the outcomes, what are the outcomes. So extract the key data, the key relevant data, and then assess the quality of the data using the statistical models with the computer, and thus analyze and combine the data to get your conclusion, which gives an overall result from all the data. That's the meta-analysis or systematic review. So this is a cartoon to mimic the study of systematic review. This one, you can think the meta, and you can think it's a research question. You know a research question, and then from the database, you collect the key data from different studies, and then use the suitable statistical model to get the overall conclusion of the study. So this is the meta-analysis or the systematic review. So this one can give you a better idea and give you a higher statistical power and help you to know better about your study. So a quick review for the advantage of the systematic review. The results can be generalized to a larger population. The precision and the accuracy of the estimates can be improved as more data are used because you combine different studies, so more data, they are more reliable. This in turn may increase the statistical power to detect an effect. And inconsistency of results across studies can be quantified and analysis. So if you read enough paper, you may find that based on one topic, maybe different groups may have different conclusions. When you combine more data, then you have your own judgment to conclude that which one is more reliable. So for those inconsistent data, when you do the systematic review or meta-analysis, you can analyze or quantify this data to get to have your own judgment for them. And also, hypothesis testing can be applied on summary estimates. And moderators can be included explain variation between studies. You know why they have different data, which one may have their disadvantages, how they get different conclusions, and the presence of publication bias can be investigated. So this is the advantage of the meta-analysis and systematic reviews. So from that already we know different types, all the types of the systematic review. Let's have a quick review. Oh, sorry. So a quick review for the study today. We learned what is medical research, right? And we described different types of medical research, including the basic research, applied research, and translational research. And also, we learned about different specific detailed types of the medical research, including the in vitro study, animal study, case reports, case series, and the case control studies, the cohort studies, and the randomized control studies, and the systematic reviews and the meta-analysis. After this class, I hope you have a general idea about the medical research. Okay, that's all for today's class. Thank you so much. If you have any questions, you can leave me a message or send me email. I will respond to each email as soon as possible. Okay, thank you so much. That's all for today's class.
Thank you. Have a nice day.
Okay, see you.